So I know there is a time constraint, and that's why I might be rushing through some of the slides. I and of course I don't know every one of you, but I assume some basic knowledge of biology in this presentation, which I hope that most of the audience has already. So just a quick introduction to eukaryotes. Eukaryotes, basically, as we all know, is a group which has creatures from animal kingdom, plants and fungus, and some other types of creatures as well. And they have cells which contain nucleus, and that's precisely the formal definition of eukaryotes. Now, that's a formal definition, but as I said, I tend to look at things from a different perspective. So, for this talk, I would like you all to forget cell has a nucleus. I mean, it does have a nucleus, eukaryotic cell, but I would like to focus on some other things of eukaryotic cell. So, the way I see it is, I like to, when someone says that this is eukaryotes, this is prokaryotes, and I immediately ask what really is the difference. And when I mean difference, what sort of vibes do I get? What sort of feel I get when I look at a eukaryotic cell as opposed to a bacterial cell? What I see really is, what I am really struck by is the complex architecture of eukaryotic cell. I see it way more complex than a bacterial cell. And of course, eukaryotic cell has mitochondria, uh, and this is almost an established fact that all eukaryotes have mitochondria. There are some very rare exceptions that people will like to point out, but let's take it as a fact that eukaryotic cell have a mitochondria in them. No need to really explain what a mitochondrion is, but I would like to stress that the mitochondria have their own genomes, which we call mitochondrial DNA or mtDNA, which also you might know. And this the circular diagram is what it's a representation of mitochondrial DNA. So, like bacteria, it has circular DNA, and it is also an established fact that the mitochondria have bacterial ancestry. So long time ago, more than 2 billion years ago, the first bacteria, sorry, not the first bacteria, a bacteria entered a eukaryotic cell. That's how the theory goes. But it is very well established that from a bacteria, mitochondria evolved, basically. Or mitochondria, what we see now, has an ancestry of a bacterium. So circular genome, it has certain genes which do specific functions, specific protein synthesis. In this whole discussions, we will be mostly focusing on mutations of mitochondrial DNA. So just as bacteria or any other organism, mitochondria also undergo replication, especially I'm talking about mitochondrial DNA here. The DNA replicates, and like any genome which replicates, there is a chance that in the process it acquires mutations when the replication happens over generations. But there is one specific thing about mitochondria or mitochondrial DNA. That is that because it is confined inside a cell, it is exposed to the reactive oxygen species, which is generated during the process of energy generation inside cell, or rather inside mitochondria. So mitochondrial DNA are exposed to that reactive oxygen species, which is what makes them more prone to mutations. And of course, mutations are also because of aging of mitochondria and aging of cell, as we will see later. So the basic problem of today's talk is mutations are fine. If there are few mutations here and there, it's just fine. Every organism, every cell can deal with that. But the problem happens when the harmful mutations, the deleterious mutations, they accumulate inside an organism. So when there is no recombination, which is what most prokaryotes divide, not via sexual means, but by asexual means. So that means there is no recombination. And if there is no recombination and the mutations keep on accumulating, it will lead a species towards extinction. And this particular law is called Muller's ratchet, which you might have heard already. So mutation accumulation is no good. Harmful mutation accumulations are not to be allowed for a species to continue to exist. So in mitochondria also, those mutations of mitochondrial DNA need to be managed. The question is, how do we manage mitochondrial DNA mutation accumulation, or how does nature manage it? So here is a scheme of things that I have proposed. So this is been published in my paper in Biosystems last year. So what I call is pretty much nature's mechanism of management of mutation in mitochondrial DNA. And I've given this a name: split, destroy, rejuvenate cycle. So let's look at it this way. You have a container. Just forget what is the container. Any container which has a number of mitochondrial DNA, the genomes in there. You can see the circles which represent mitochondrial DNA. Now the container ages. As the time passes, the container ages. Uh, Inside the container, replication is going on. So mitochondrial DNA are replicating. And in the process of replication, aging, and other factors, there are mutations happening to certain mitochondrial DNA genomes. So you see some red circles in there. The red circles basically represent mutations. So this is just a representation. It doesn't mean there are 4 genomes and one of them is mutated like that. It's just a representation that a fraction of all mitochondrial DNA that keep on replicating themselves acquire mutations, harmful mutations. So what happens to the container thereon is the container splits. Now this is all conceptual. So this is a model or a workflow, a generic strategy, a generic policy through which eukaryotes manage mitochondrial mutation accumulation. So when the container splits, it has few containers which carry more mutations and some others which carry less mutations. So the ones which carry more mutations, they are led along the pathway of destruction. So the way the scheme of things work in nature is they are directed towards destruction. So that's why we call it destroy. And the ones which are which carry less mutations are carried along the path of rejuvenation, which somehow magically uh, rejuvenates or makes the container young again, and the whole cycle keeps on repeating. That's the broad scheme of things how mitochondrial mutations are managed, and I call it split, destroy, rejuvenate cycle. So if there is one thing that I want everyone to take from this talk, it's just this slide. You can forget pretty much everything of what I'm going to talk about, but remember this split, destroy, rejuvenate cycle. Um, and and we'll see in details for the rest of the talk how this scheme works in different containers. So you might be surprised to hear this strange term container. It's a very everyday term. It's not a scientific term, but I prefer to use this because it explains, it pretty much tells what it is. So my first container is a mitochondrion, which is a very obvious thing. So a mitochondrion contains mitochondrial DNA. And the next higher level container I will talk about is a cell, which contains a number of mitochondria, which in turn contains a number of mitochondrial DNA. Then we have organs, which are made up of a number of cells, and then the organism. I won't talk much about organ today. Our talk will be mostly focusing on mitochondrion, cell, and organism as containers. So these are containers a level up every time. So mitochondrion is the innermost. Then we have cell. Then we have organism. So, just a brief introduction to mitochondrion as a core container. I mean, we all know what mitochondria is. We've been reading it from school times. But as I said, what I want in today's talk is a different perspective. So, having a look at that from a different perspective. Uh, if I ask any layman even what is a mitochondria, he might come up and say mitochondria are the powerhouse of the cell, which is a very common thing. So which means it carries out the function of respiration here. But equally important, if not more important, I consider metabolites synthesis inside mitochondria one of the most important features of mitochondria, which is basically the very core of biomass generation in living systems in eukaryotes. That's the start, that's the pillar. It provides some PC cycle intermediates which are then used to biomass generation in cell and in organism. And for that, it requires some input materials like pyruvate, fatty acids, oxygen, and some other things. Then this is mitochondrial DNA, as we have seen before. Mitochondrial DNA, like any genomes, the purpose of that is to express the genes and create proteins which are required for carrying out the functions. Now, funnily enough or strangely, mitochondrial DNA doesn't cater for all the functions that are happening inside mitochondria. So a mitochondrion rather contains say 2 to 10 mitochondrial copies in mammalian cells. The number varies across the range basically. Mitochondria are dynamic, so it's even harder to ascertain the numbers. So the number, the size and shape of mitochondria depends on the state of cycle where it is basically at that particular moment. So it undergoes a fission fusion cycle continuously almost, and you cannot determine the number perfectly and say that that mitochondria has that particular number of mitochondrial DNA copies. Um, so when we say it is a dynamic thing, uh, there is something called mitochondrial membrane potential, which is basically just the proton reservoir, the pool of protons that are present in the intramitochondrial space. I think this is all basic biology. Most of you will always know this. Um, so this membrane potential is basically a proxy for health of a mitochondria. And mutations and membrane potential, they go hand in hand. If there are more mutations of a mitochondrial DNA, it affects membrane potential, obviously, because if the genes are mutated, it means it's going to produce faulty apparatus which will not function well, and the whole electron transport chain will not work perfectly, and there will not be proper proton pumping, and that's why it will affect the membrane potential. And if there is low membrane potential, it makes the mitochondria unhealthy. That means it cannot support a growing number of mitochondrial DNA population. So we talked about fission. So after fission, you get a number of mitochondrial fragments, and if a particular fragment has low membrane potential, it's marked for destruction. So, and then there is fusion also in mitochondria, which basically is a combination of a number of mitochondria to make a network of mitochondria. And when they are in a fused state, there is a lot of exchange of materials that are inside. So exchange of metabolites, exchange of proteins, and even exchange of DNA, which basically goes on to rejuvenate the mitochondria. And fusion is also known to lead to reduction of mitochondrial mutations. So let's look at these dynamics with the split destroy rejuvenate cycle that I had mentioned before in mind, and see how that fits in the scheme of things. So we have mitochondria, which contains mitochondrial DNA, and the container ages. The container is mitochondrion here, and you get mutations. Then fission happens. So in this case, the split is actually accomplished via fission, and as a result of fission you get different fragments, and the fragment with more mutations are led to the pathway of autophagy, which in terms of in this case we call mitophagy, and the fragments with less mutations, they are driven along the path of fusion, which is the mechanism of rejuvenation in mitochondria recipe. So it kind of follows the same scheme of theme that we talked about earlier. So let's move on. I'm really aware of the time factor, so that's what I'm trying to rush through. I hope people are getting what I'm talking about. Um, is it fair to assume so far?
Absolutely. Yeah.
Okay, thank you so much for confirming. So let's move beyond mitochondria. So a cell contains, on average, 1,000 mitochondria. Now this means nothing. This figure means nothing because the number varies really, really widely, from just a handful to even hundreds of thousands of mitochondria inside one cell. It also depends on the tissue type and the state at which that particular cell is. So this number really means nothing, but just to have some number as an average number, we can assume that roughly 1,000 mitochondria per cell. And as we see, to carry out their function, they need regular supply of nutrients like pyruvate, fatty acids, oxygens. Uh, so cell, again, change of perspective. The way we have been seeing cell, I want to look at it from a different perspective. The cell, in this instance, is a captive storehouse of nutrients. So mitochondria need nutrients to function. Cell is a storehouse. It keeps the nutrients and supplies to mitochondria. Cell also is a provider of shared infrastructure and services. We know microtubule network acting structure and all that, which basically, and other features, endoplasmic reticulum, Golgi apparatus. Uh, so they are all like shared infrastructure shared by a number of mitochondria. So this is cell, again highlight on mitochondria because that's the topic of the day. But there are more mitochondria inside cell, obviously. Here we have just shown one mitochondria. So apart from these two functions that we discussed before, we have a lot of protein synthesis happening in the cell. A lot of them goes on to serve the mitochondria because mitochondria cannot create the protein, all the proteins that it requires. So a lot of protein is imported into mitochondria, and they are uh, created using nuclear genome. And so for that we need another input material, amino acids. And there is, we all know that the cell dies. There is a program cell death mechanism, apoptosis, which is also regulated by mitochondria. So mitochondria are really, really the key organelle in the cell. Now, then we are only worried about mitochondrial DNA copy replication and the mutations as a result. So if the mitochondrial DNA number keeps growing all the time, we definitely require more nutrients all the time and more space for the mitochondria to be accommodated in the cell. So obviously the easiest solution is to keep on expanding the boundaries of cell, but hang on, that is impossible. That doesn't work, and that's because of metabolic scaling. So this is there's something called metabolic scaling law. I won't go into the details of that, but it all comes down to a very basic geometric relation or laws of geometry or mathematics, which basically tells you that if you expand a balloon, It's the volume increase is much more compared to the surface area increase. And that's the reason, precisely is the reason we can't keep on uh, pushing the boundaries and expanding a cell to accommodate more mitochondria. So the only solution is cell division. So a cell has to divide to accommodate more mitochondria. So now let's look at cell as a container and how it goes on doing the division. So we have a cell and we have mitochondrial DNA. Forget there is a mitochondria also, which contains mitochondrial DNA. But the cell ages, and again the same thing happens. More mitochondrial DNA through replication, more mutations. Now cell divides via mitosis, and so, I mean, it creates a number of different population of cells, but I'm conceptually dividing into two types of cells. So this is again a conceptual split. If you are thinking in terms of my earlier slide where I talk about split, destroy, rejuvenate, this is not exactly a physical split. It is a conceptual split. So some population of cells become somatic cells and some population become germ cells, very few actually. So the somatic cells are ultimately led to the pathway of apoptosis. They carry out all the day-to-day activities and all that, but their destiny is death, basically, in the end. Whereas the germ cells, they are carried forward and into the cycle, and that's how eukaryotic cell works, basically. So there is a cell cycle which creates more cells. Cell division is governed by the processes inside the cell cycle. Uh, so now we know there are two things: mitochondrial DNA replication, and there is a cell cycle. So both are cycles or uh, iterative processes happening inside our bodies. So it is said that mitochondrial DNA replication is independent of cell cycles. It doesn't follow the cell cycle. However, there are things called relaxed replications, which are truly independent of cell cycles, and there is strict replications, which actually tend to follow the cell cycle pattern. And there's also synchronization of fission and mitochondrial fission and cell division. So how mitochondria kind of has influence on cell cycle? This is a very active area of research. We don't know everything yet, but these are the points from very latest in the literature, and this gives us some clue as to why mitochondria might be the most important thing in cell cycle regulation. So we have these different phases of cell cycle. So I'm trying to get hold of the cursor, mouse. Yeah. So at this G1 and S phase boundary, at this transition, there is a checkpoint. We know that as G1 to S checkpoint, which is where there is a decision made on to whether to proceed with cell division or not. So that's a go, no go decision point. And at that point, we know now that mitochondria fuse, and in fact, they are in a hyperfused state. Now, what is the implication of all that? We still don't know yet fully, but definitely that thing happens there at that stage. I mean, to have fun, I like having fun with science. I say that mitochondria, they organize a meeting and they fuse together and they have a great meeting within themselves to make a go no go decision. But that's the fun side. That's not the real thing. Um, then there is this second phase, and another phase transition between G2 and M phase. At this point, actually, all the protein import regulation happens. You know how mitochondria requires protein to be imported from the cytoplasm into mitochondria. Uh, but that That entry gate is built via this complex Tom-6 complex, and that entry gate is actually constructed right at that stage, and it regulates how much of mitochondrial biogenesis should take place during cell cycle. So there is definitely regulation happening. So there's a bit of synchronization, coordination between mitochondria and cell cycle there. And this is even more interesting, that the amount of mitochondria that is distributed in the daughter cells, it has been found that they are proportion to the cytoplasmic volume of the daughter cells. Uh, and even in budding yeast, which propagate via buds, creation of bud, so it takes stock of the volume of the bud, and depending on the volume available in the bud, it accordingly transfers the number of mitochondria to the buds. So what it tells me is that um, there is a stock take happening there. That okay, we've got daughter cells, so we've got more cells now to occupy mitochondria, but it doesn't mean that mitochondria go randomly or any way. There is a stock take happening. How much resource is possibly going to be there for future of mitochondrial survival, and accordingly the quantity of mitochondria are distributed. So there is a lot more happening than what we know if we just focus on cell as a separate entity or the basic unit of life, as we say. There's a lot more happening there. Now, of course, cell divisions provide a new refuge for growing mitochondrial population, so which is growing all the time because mitochondrial DNA is replicating um, almost all the time. So it requires new places to be accommodated. But can cell division go forever? It can only if the environment supports a supply of nutrients, limitless supply of nutrients, which is definitely not the case always. And it's definitely not the case in land environment in which we live, and most of the land animals live. They are in an environment which by default is deprived of nutrients. So there's a lot of things goes on to make those to ensure the availability of nutrients for eukaryotic organism and cells and mitochondria. So this leads to a situation where if you only rely on cell division, all the daughter cells are going to be destroyed, or they'll be wiped out because of the harshness of the environment, and with them all the mitochondria also will vanish. So there is a need for a rejuvenation pathway here. So how do you think the rejuvenation happens? It is by adding the feature of the phenomenon of sex or sexual reproduction. So we'll come to this slide again later, but that's what I'm just basically expanding this line here. So there is the phenomenon of sex, which actually rejuvenates the container and the cycle goes on. So to understand the history, so this is where we focus a lot on evolution, which means we go back, we keep going back in time and try to understand how things might have been long time ago, millions of years ago, and how they are now. So how the evolution has taken place. So unicellular creatures provide us with a nice uh, test cases to study and understand various evolutionary features, because we all evolved from unicellular creatures at the end of the day. So let's see how they handle sex, uh, unicellular creatures. So we can study diatoms, we can study ciliates or green alga. They all exhibit various features of sex. We can get a lot of clues from that. But for today, I'm just focusing on ciliates, which are very interesting. They have two nuclei basically, one macronucleus and one micronucleus. So macronucleus is like a day-to-day nucleus, a working nucleus for them. And that's why I tend to equate that with somatic cells in our bodies. And micronucleus is a reserve kind of a nucleus which is kept there quietly, uh, and not participating in day-to-day functions. So that's why I call that equivalent to germ cells, basically. So when there are plenty of nutrients available, CPFs are just live their happy life, dividing vegetatively via cell division, and there are plenty of nutrients available, so no problems. But when there is less nutrients available in the environment, that's when they go for sex to rejuvenate themselves, basically. And the way it happens is that the cell divides via meiosis, and then there is fusion happens between cells, ciliate cells, and uh, there is a new micronuclei generated by fusion of micronucleus, two micronuclei, one which is stationary and the other which one is migratory. They fuse together and create new micronucleus. And from that micronucleus, we get a macronucleus for day-to-day functioning. And that the way it works is that the ribosomal DNA of micronucleus is copied over and over again, and thousands of copies are created and put in the macronucleus, and then it is back to the vegetative growth stage then. So back to normal cell divisions, and that cycle keeps on happening. It's only at the time when there is less nutrients that they go for sex. Same with diatoms, same with green algae. They all follow the pathway of sex at the time of when there is less nitrogen availability, less nutrients in general available. So the idea, the how the sex came about initially and evolved over time is we have normal cell division when nutrients are available. When there is a nutrient deprived conditions, the creatures go for sex, basically. And while when it rejuvenates via sex, it falls back on the sacred or the least eroded genetic material. For example, the micronucleus, which is not used for day-to-day purposes. They're kept special quietly in a corner for use when required, and that use is when during sex. And the differentiated genetic material, for example, the macronucleus in ciliates, they carry out the business as usual. So we see there is a soma germ differentiation happening in nucleus in ciliates. And there is another type of split happening. This is a conceptual split again, not a physical split. So there is that split of soma germ line happening, and there is another split which we call mating type split, basically. So the stationary ones are like females, and the migratory micronuclei are like males. So there are two different mating types required. So that's another conceptual um, split which is required for the process of sex. This is just a history of soma germline differentiation. It started with cyanobacteria. So cyanobacteria, which were present more than 3 billion years ago, they had these features, very primitive features of soma germline differentiation. I will just skip this slide because we have a lot to cover. But this is very interesting, really, if anyone wants to check out the history of soma germline differentiation, how it evolved with cyanobacteria. So coming back to that slide that I skipped earlier, so it's basically the process, the split destroy rejuvenate cycle in operation using sex. So sex is basically causing the rejuvenation of cell. So we have meiosis, meiotic divisions, we get female egg cells, and from similar path comes the male sperm cells, and then they fuse, and then they make a new container, fresh container. But mind you, the container is fresh, but the mitochondrial DNA inside is all coming from the female side. which is what we call uniparental inheritance or maternal inheritance. And all the old mitochondrial DNA more mutants they get destroyed via mitophagi. So sex fundamentally is a quality control mechanism for as far as mitochondrial DNA is concerned. So if I'm looking from a perspective of mitochondria, the sex of cell as well as the sex of organisms are basically just purifying mechanisms or a mechanism to keep the mitochondrial DNA as pure as possible. So there are various processes that are happening during fertilization development, like purifying selection, mitochondrial bottleneck, mother's curse. You might have heard about that more mutations are passed along the male line because they are going to be destroyed eventually. And this all goes together with uniparental inheritance in eukaryotes in general, and in most cases it is maternal inheritance. As Dr. Mohanty said, mitochondria or mitochondrial DNA are passed along the female lines, basically. So this is the representation of split destroy cycle, split destroy rejuvenation cycle in organisms. So you have a organism as a container which has mitochondrial DNA. Forget all other boundaries. Um, the container ages, the person ages basically. Uh, at maturity, they give rise to offspring. So you can have a male offspring or a female offspring. As I said, the phenomenon of mother's curse basically usually passes more mutations to the male side and less mutations to the female side. Then they all become adults and then they perform the act of sexual reproduction. Well, not really coming from same mother because they will be brothers and sisters, but we are talking of population here in general. And this is just a representation, otherwise the diagram will become very complicated. So sexual reproduction happens, and the body is rejuvenated in the form of a new, fresh organism, basically a new individual organism which carries the mitochondrial DNA from the female side. And everyone ages and dies. So death is a destruction mechanism at the organism cell. So, as I said, it's all about different perspective. There's nothing new in this presentation, no new facts. It's just the perspective which is new. Uh, Dr. Monti represents stem cell facility, so I thought I might just add this. It will be a little bit interesting. So here we are talking about organs, but this is just a side note. So we can think of organ stem cells as partial rejuvenation pathways in the soma line. We know that the soma line is meant for destruction, eventual destruction, but just to maybe extend the life a little bit, there is partial rejuvenation happening along the soma line via the stem cells. And funnily enough, again, it is found that in mammals, stem-like cells, they usually inherit the young mitochondria, whereas old mature mitochondria are passed along differentiated cell line. So it all makes sense if you look, I think, from the point of mitochondrial DNA. It's like, you want to have the young ones uh, to be taken care and not engaged in day-to-day tasks. No child labor, please, in the case of mitochondria. So all the oxidative phosphorylation, the energy generation mechanism which requires mitochondria to work, happens in differentiated cells, whereas in pluripotent cells and other stem cells, usually they give mitochondria rest and use the energy generation mechanism of the cell infrastructure via glycolytic pathway. So this is again nature's mechanism of basically keeping the the rejuvenation pathway as sacred as possible, as untouched as possible, as least eroded as possible. All right, so this is a very busy slide. Basically, it's the same thing that I've spoken about in the previous slides. But this basically sums it all up at the organism level. Uh, so, an individual circle is a representation of a cell. Uh, if it's fully filled by the same color, that's what we call homoplasmy. I hope that people know what homoplasmy is. It's when you have mitochondrial DNA of the same variant, basically. All the copies, if all the copies are of the same variant, it is homoplasmy. If there are a number of different variants because of mutations, so differently mutated mitochondrial DNA, they come together, we have a heteroplasmic state. So to start with, oocyte is heteroplasmic. Previously, it was thought that it's homoplasmic, but that's not the case. And then it goes through these processes of purifying selection, genetic bottleneck, um, and then massive replication happens. This mature stage oocyte has as many as 2 lakh mitochondrial DNA copies at this time, ready for fertilization, basically. And here comes the poor male with its sperm cells with very few mitochondrial DNA copies. And it comes and contributes in creation of the new cell after fertilization. So, and again, as I said, and as Dr. Mohanty says, most of the mitochondrial DNA are coming from this line and they're going through this line, basically. Uh, Even if some mitochondrial DNA from the paternal side survive this stage, they are soon eliminated. So, sorry, male folks, that's where your role ends. It's all about female. I mean, we talk of Vansh, but real Vansh carriers are females. Uh, so, and then cell division happens. So it goes through various stages of embryogenesis. And there is another bottleneck here, which kind of leads towards homoplasmy. And that I see as, preparing a very fresh organism's prototype, giving it with all the goodness. And here you are, ready for facing the life, go on, grow. And because we know that as it grows, it will acquire mutations. So mitochondrial DNA will acquire mutations inside. Uh, as it grows to fetal cell, as that grows to tissue cells, more mutations will happen. And the ultimate fate in most cases is cancer cell, where mostly the mutants kind of take over and reach a near homoplasmic state with mutations, and that's really dangerous, and we all know that that leads to cancer. So I've tried to include that all in this picture. And so we have talked about various processes that help reduce the effects of accumulation of mutations in mitochondrial DNA. So how about let's look at how mitochondrial DNA might have evolved over time. So it's said that the alpha proteobacteria entered the eukaryotic cell roughly 2 billion years ago. So 2 billion years ago, which is like 2000 million years ago. Uh, and they have the mitochondria have the lineage of alpha proteobacteria. Now, there are alpha proteobacteria currently also available, so they are living now. Maybe not the same species which give rise to mitochondria, but some species. So this indicates a typical genome of alpha proteobacteria as we see now. If you look at the size of that genome, it's 1.3 mega base pairs. And the size of mitochondrial genome is just 16.5 kilobase pairs. So it's much reduced. So massive store of genes to all the works that alpha proteobacteria needs to do, but mitochondria only a handful of genes in it and very less size. That is evolution because it helps. Carrying smaller genome is already always helpful. It helps number one in faster replication because there is less genetic load, and it also helps with keeping it pure, so having less mutations along the way. The bigger genome that gets duplicated or replicated has more chances of mutations happening or errors happening in copying mechanism. So definitely mitochondria are smarter things. They have acquired, they have been along a pathway where they could reduce their genome size massively. But the question is, where does those genes go? Um, so where does those genes go? Which, if alpha proteobacteria had so many genes and mitochondria have such less amount of genes, so they have gone into nucleus. In fact, so evolutionarily, there are mechanisms where transfer happens from mitochondria. If we are talking of plants, the transfer also happens from chloroplast to nucleus. But here we are focusing on mitochondria. So mitochondrial genome has been transferring its genes to the nucleus. And look at the poor nucleus. It's massive. The nuclear genome size is massive. And, and very small fraction of it is useful for protein synthesis as we know now. But there has been a mechanism of transfer of genes from mitochondria to nucleus, and those genes, pseudogenes rather, they are called numites, NUMT, which is short for pseudogenes of mitochondrial origin, basically. Which so you have nuclear genes, but they are of mitochondrial origin, and they form part of the what we call junk DNA of nucleus. Are they really junk? We don't know. There could be things for which they are used. We simply don't know at this stage. But that's how mitochondrial DNA evolved over, say, 2 billion years. Um, this is very complex, and I apologize for this complex diagram. Just to get your way through this figure, I'll ask you to focus on the center, very center, where you see a mitochondrial DNA. So this is a representation of how this whole process of mutation accumulation and evolution is working, basically. So at the mitochondrial level, you have mitochondrial DNA, which has a tendency to create more copies of itself. It wants to live forever. Whether we come from ocean to land, it still wants to be part of life in there. And if we eventually move to Mars or some other extraterrestrial body, it will still be there. That's what it wants, basically. So a mitochondrial DNA copies. For copying, it requires materials, it requires energy. So this dark green color is the material line, how it acquires materials. And this is the energy line, how it acquires energy that's required for this mitochondrial DNA copy or replication. And as a result of replication, you have more mitochondrial DNA copies, and you also have mutations, unfortunately. You have more mitochondrial DNA copies, you need more space for them to reside. You have fission of mitochondria, which creates more fragments. You have more mitochondria, you have cell division, you have more cells. Differentiation, you have multicellular organisms. And each of these containers, as we have seen, they age. They are not fresh all along. They aged because they face the environment, and there is weathering and erosion happening because of interaction with the environment. So they age, which causes even more mutations. So this red one is the mutation line, or rather mutation remediation or mutation management line. So the copies have mutations at the level of mitochondria. The mutations are managed using fusion and mitophagy. At the level of cell, mutations are managed using meiosis, syngamy, and apoptosis, cell death. At the level of organism, mutations are managed via sex and death. And at the level of species, which we haven't touched, which is where I want to do more research in future, the mutations are managed via gene transfer to nucleus. See how smart mitochondria are. They keep the pure ones with them and the rubbish ones go to nucleus. And so that's basically how I see evolution of mitochondria happening. If you, this is the last slide. So if I apologize if I've gone past the time, but this is the last one, and I won't take much time with this. So imagine a free living proto mitochondria like around 2.5 million years ago. This green line is the oxygen level of atmosphere. So this is say 2.5 billion years ago. This is present right now. Of course, this is schematic. It doesn't mean that this happened at 1 billion year mark. Or, no, nothing like that. But this is very much a hypothetical drawing. I just want to understand more how protomitochondria evolved from being free living by itself to occupying human body or all eukaryotes body and living inside cell. So what we have now is we have high protein to RNA ratio. So it started with more RNA than protein. We now have more protein, definitely more protein to RNA ratio. It has a high nitrogen to phosphorus ratio. It has high GC content. Now this is very important. G and C, these nucleotides are, as you may know, are kind of expensive in biological terms. They are expensive nucleotides and they are most stable nucleotides. So again, see how it tends to corner the best of the things itself. So it has high GC content, whereas nuclear genome, I think there is a lot of AT in nuclear genome. And of course, it is dependent on cell for support of machinery accessories and it has a control over cellular processes. That's another topic of discussion. I have written another paper on that basically, which highlights the importance of mitochondria, or rather centrality of mitochondria in eukaryote biology. So, and that's how I see the evolution taking place. But this is a very hypothetical thing. Please don't quote me on this. This is a hypothesis at this stage that how mitochondria physically evolved to the present state. Thank you so much for listening to me. And if you have any questions, I mean, I don't know how question answers will work today. Dr. Monty will advise. I'm happy to take questions. And if you want to contact me for anything, this is my private email address, abdeonath at hotmail.com. So mark the two D's there. So it's ABDEONATH at hotmail.com. This is my private email address, which I have access everywhere basically. So yeah, thank you very much. And if there are any questions, I'm happy to take.
Thank you so much, Dr. Abhijit. I think I would request everyone to post any questions here. Um, uh, so before speaking, I would say thank you so much for at least changing my perspective. Like I never thought that this tiny mitochondria, which we call a powerhouse, is really a smart. Very, very smart. I think we should learn from here, from aging to sex, rejuvenation, and like we can really say that you have rightly used the word smart. So we should learn from this mitochondria also to our daily life and in my scientific endeavors. I think I have gained the maximum. I have few questions also, but before that, I will give a platform to the students. Pranshu, would you like to speak? You can unmute and speak. I think he's not able to hear. So here's a question. What can be the implication of insertion of foreign mitochondria in damaged cells? For example, multiple mitochondria from different sources in a single cell. So he's asking like what will be the scenario if
Right.
mitochondria are getting from different donors into one cell. I hope I read his question properly. Yes.
Right. All right. Okay. So who asked this question?
He is a PhD student working on 3D bioprinting. I think he's
Yeah.
Pranshu.
What's the name? Sorry. What's the name? I
E
missed it.
Pranshu. Pranshu.
Oh, Pranshu. Pranshu.
Pranshu.
Okay.
Pranshu.
Yeah. Thank you, Pranshu, for this question. It's definitely very interesting question and very important question, given what I talked about mitochondria. Um, Now, I'm not from the medicinal background, so I'm not. My background is not medical science. So, from the medical science perspective, I won't be able to give you an answer, unfortunately. But I have read a lot about mitochondrial transfer. It happens naturally, also in between different tissues. There is mitochondrial transfer happening. Uh, Even in cancer stages also mitochondrial transfer are happening. And I heard about mitochondrial therapy. Uh, so definitely it looks like there is a possibility of having healthy mitochondria injected into damaged tissue to fix the tissues. There's definitely a possibility there. Um, yeah, that's all I can say at this stage. I'm sorry, I'm not from medical science background, so yeah. I
Uh,
hope I could
thank
do justice to the question.
I think very well, yes. There
Yeah.
are now strategies where people are purifying the mitochondria and now they are giving the cocktail of mitochondria in certain diseases, which heart is one of them. So we just to tell you, Dr. Abhijit, we have here the faculty, some students, PhD, postdocs, and also some like graduate level, BSc level students, and they have all said, thank you so much for this wonderful session and very interesting perspective. I'm just reading their messages on the chat box. Yeah. So any
Thank
further?
you.
Meenakshi, you have
Thank
raised
you
the
so
hand.
much.
Yeah. Meenakshi, would you like to speak? I saw you raising a hand. Okay. Then Shruti, would you like to speak or shall I read your question? So she's saying, does nutrient supply affect the membrane potential of the mitochondria? She is Shruti, a PhD student. Yes.
Thank you, Shruti. Yeah, of course, nutrient supply is a big thing. Mitochondria do require nutrient supply to carry out the functions of PCA cycle and electron transport chain, which are the very fundamental thing of their existence and our existence in general. There has to be nutrients supplied all the time to mitochondria. So if the cell fails to do that, that's when mitochondria trigger the apoptosis button, and uh, it leads to death of cell, basically programmed cell death.
Mhm. And there's no one else right now. So, Abhijit, I have a very vague question. Please excuse me if it looks very redundant. So,
No,
as you said that mitochondria from two billion years ago, like it started coming into the cytoplasm of the cell, try to maneuver its way, giving out some of the junk DNA to the nucleus, and then maintaining the good nucleic material for itself. That means this process is continuing. So can we predict that what is going to happen in future? As I said, this process is again a dynamic. Can we predict in which direction this mitochondria is going to do?
Uh, this is brilliant question. This is brilliant question. Actually, this is where basically I might have to go into a bit of science fiction area
Yes, I think so because
if we
it,
keep thinking too much.
it is really a smart organelle. Like we always think it as a powerhouse and then we forget. But I think it's really a smart. We need
Yeah,
to follow it and learn from it. Like it's like a nature which is teaching you, sort of that.
yeah, absolutely, absolutely. And to give some response to what you asked here,
Mhm.
which is sorry, I'm trying to recollect my thoughts here. So you want to know if what will happen in future. So where will this evolution take in future? Okay, that's very interesting. I mean, no one knows for sure what's going to happen in future. It depends on a lot on. So how I see this whole eukaryotic life mechanism working is the central purpose seems to be growth. Like we have to move to uncharted territories. So there are two philosophical aspects of life. One is
Mm-hmm.
survival and another is growth. So you
Mm-hmm.
have to survive to be able to grow, but you
Mm-hmm.
just can't rest by surviving. You have to still take a step further. That's why we have legs, or we are motile in a sense that we are not meant to be, rooted like plants. If I'm talking of animals. So we also need to continuously evolve, I guess. Uh, and I mean, this definitely takes me to the territory where it's not really that much of science. It's a lot of speculation that I can make some educated scientific guesses here at best. But this is definitely not science, and I think that as an organism or as a species, or we love to call ourselves the most advanced species. If we are truly most advanced, our job, our purpose of life is to keep going forward. So we have to take the mitochondria to uncharted territories. So it has new territories where it can survive and thrive and flourish and propagate itself, proliferate. So I think of ourselves as just container of mitochondria, basically. So we have to take the mitochondria along, and that's why we have the function of motility, and we've got a lot of gray matter in our brains, which helps. I mean, if you look, even neurodegenerative diseases, there's a lot of control. It's got a lot to do with mitochondria. So you talk of every disease basically. Parkinson's disease, Alzheimer's disease, or even cancer, heart diseases. They all lead to mitochondria. They all lead to mitochondrial dysfunction somehow, some way or the other. So it is telling me that's important, and we need to look after the health of mitochondria. Make sure we go to new places to give them new space, and organize the resources for feeding mitochondria, basically. And if we don't do that, science fiction stuff here, in don't quote me on this, we will be extinct.
I think you very well said. I think I agree with you 100%, and it's a very challenging and interesting area to keep moving forward. So, just to give an example, we have two more questions. Before that, we did some initial studies, and we thought that when we are bringing stem cells near damaged cells, so somehow the cell is only giving very new and fresh mitochondria to the dying cells. The old and invalid mitochondria are being retained. So there is a lot of cross-track smartness, what you say, coordinated process going on between the mitochondria and the cell. We need to dwell upon that if we want to target the future diseases which are going to plague us, like diabetes, Parkinson, Alzheimer, and all these things. Very well said.
Yeah,
Yeah.
definitely. Yeah.
So we have two more queries. Dipanshu, would you like to speak? So Dipanshu is asking, is there any preferential metabolic pathway that mitochondria rely on during fission and fusion?
Thank you, Dipanshu, for this question. So let me read it again to register properly. Is there any preferential metabolic pathway that mitochondria rely on during fission or fusion? Um, I don't know, to be frank. I definitely don't know whether it follows the pathway of fatty acid oxidation, or it follows the pyruvate pathway more. I know that during cancer, it blocks the pyruvate pathway and prefers fatty acid oxidation pathway. Especially for fission or fusion, I'm not aware that it follows any metabolic pathway in preference. But if you have understanding of it, if you have read something about it, I would like to know. I mean, please feel free to enlighten.
So I need to study this. Actually, I've not read about it in detail, but yes, I'll see if I can find anything.
Sure, sure. I would like to know
So
definitely.
I have another question. What was
Yep.
the evolutionary basis for not having recombination in mitochondrial DNA?
Right. Okay. Uh, this is a very challenging question. Evolutionary basis for not having recombination. Um, so, I mean, recombination requires it to get another type of mitochondrial DNA genome, I guess, or get something. So I don't know recombination, like even in bacteria, recombinations are not the common things, but so they mostly tend to replicate their copies, and that's how they proliferate basically via fission mostly. So yeah, I really don't know what could be a possible evolutionary basis for not having recombination. Uh, but yeah, that's a good observation that they don't follow recombination. It could be that they have basically accepted the destiny of living inside cells, and that's how they evolved through proliferation by replication only.
Thank you, sir.
No problems.
We have last question from Yashvi. Yeah. So, Abhijit, would you like to read and give answer to that? Is above the Panshu query? Yeah, in the chat box.
Okay, let me look at it. Yashvi Sharma, yes.
Yes.
So, what is your opinion about the dynamics of stem cell derived mitochondria being different from somatic or germline mitochondria? So, as I said in one of my slides, that as far as I know, the stem cells have more young mitochondria compared to somatic and differentiated cells. So, so they have young mitochondria. They are mostly to be taken care of, looked after, not used in cellular functioning, and so that's all I can say about dynamics of um, mitochondria which reside in stem cell. I won't call stem cell derived mitochondria because mitochondria are not derived from cells. I, from the perspective that I believe in, I think cells service mitochondria more than the other way around. So that's the traditional perspective that cells make use of mitochondria for energy generation, but I don't subscribe to that view. I feel that mitochondria have their own purpose, and cell serves mitochondria, supplies nutrients, and harbors nutrients for consumption of mitochondria, and also supplies proteins via gene expression through their own ribosomes and through nuclear genome, basically. I'm not sure if I have answered your question, Yashvi, but definitely we can have email chats or other chats about that.
Thank you so much. Any further questions? Yeah. So she's right that she can email you if she has more questions about it. I think at the end, I would like to take the pleasure of thanking you, Dr. Abhijit. Thank you so much for igniting and exciting us and our brain to think more about mitochondria. And I really like the way you have explained your perspective. Maybe it's hypothetical and theory, but it has really put a seed of thought in our mind to think over it and then consider this mitochondria a really a powerhouse and a star and a champion. Thank you so much, Dr. Abhijit. Thank you so much for coming and giving a talk. Yes.
Thank you for inviting me, and thank you everyone for listening so keenly to my talk. Thank you everyone, and you all have a good time, good Monday, and rest of the week.
Thank you so much. Thank you. Bye.
Thank you. Bye.